Hello, everyone, and welcome to today's broadcast, Cilia, Abnormalities and Developmental Defects in Lowe Syndrome, presented by Dr. R. Claudio Aguilar, Associate Professor and Assistant Head, Department of Biological Sciences, Purdue University. I am Xavier Gutierrez of LabRoots, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. Our speaker will respond to your questions via email. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Aguilar. I will now turn the presentation over to him.
Thank you, everyone, for attending to this talk. So as Xavier said, I'm going to be talking about the Cilia abnormalities and developmental defects in Lowe syndrome. But before going into that, let me tell you a little bit about our lab. Our lab is interested in vesicle trafficking and protein sorting in health and disease. And we're particularly interested in many vesicle trafficking events, but specifically endocytosis and recycling is what occupies more of our time. We are interested in this because, as you may know, endocytosis is very important for the distribution of proteins around the cell, but also to control signaling events within the cell. So that is what makes this process extremely important for cellular functions such as cell growth and proliferation, cell movements, and some others. Here I'm giving you some examples to try to highlight how important this process is, as endocytosis absolutely requires for nutrient uptake, bacteria phagocytosis, synaptic vesicle recycling. However, we are not only interested in understanding how endocytosis contributes to these processes, but we also want to understand what happens when some of these elements of the machinery that makes endocytosis run are not working properly. In that case, we have, for example, cell proliferation going out of control, and therefore we have cancer, cell movements that are not really orchestrated with the rest of other processes that are going on in the embryo, and therefore we get developmental diseases. So it's obvious to say that all what it has to do with understanding the process of endocytosis and its role in proliferation and cell movement is our basic research area, while what it has to do with the understanding of how what happens when the machinery is broken has to do with our disease-oriented research. And of course, we want to understand how these processes contribute to the disease-oriented status. So let me tell you a little bit more about the specific endocytic events that we're interested in. We're interested in Clattering-mediated endocytosis. As you know, is the most famous of all the endocytic events or endocytic processes studied. It's very easy to recognize by the soccer ball looking like structures that characterize the Clattering-coated vesicles. And it all initiates with the receptor being bound by the ligand and the immediate recruitment of endocytic machinery, which in turn recruit the most famous coat in vesicle trafficking, which is Clattering. So two of those players involved in the recruitment of Clattering are called epsin and the very famous Clattering-associated adaptor complexes, or APs. Some years ago, a dozen years ago, we figured out that the epsin molecule was not only involved in the recognition of cargo and bringing in Clattering to the plasma membrane, but also involved in regulators, in the regulation of cell polarity by interacting with Rho GTPase gap-regulating proteins. And this is also one of the areas in which we are very interested in understanding how signaling is controlled by endocytosis via the direct interaction of the endocytic machinery with these Rho gaps, but also by the control of the receptor dynamics in the plasma membrane. And this is the focus of our research. This complex is the focus of our research. And here I give you an idea of the different areas that we cover. We are interested in understanding ligand binding and how the induction of endocytosis occur, how the endocytic machinery recognizes its cargo, the membrane proteins that has to be internalized, and how this connection with Rho gaps are important for cell division, cancer, and developmental diseases. For example, in terms of understanding ligand binding and induction of endocytosis, we are very interested in using this knowledge, this expertise of many years, in the development of novel therapeutics for bladder cancer. And bladder cancer is very special in the sense that the urothelium is the epithelium that covers the bladder, is where the tumor cells arise, and while the normal cells are protected from the content of the bladder, which is the urine, by glycosaminoglycan layers and tight junctions and whatnot. Tumor cells usually are less differentiated. They protrude towards the lumen of the bladder, and then therefore they are more accessible for therapeutics. Now the problem is that in the bladder, there's a constant influx of urine, including urea, of course, main component, which is a challenge for any therapeutic that is added to the bladder. And also, of course, once the bladder is distended by the addition of the agent, of the chemo, the bladder senses that as it needs to void, and therefore it needs to eliminate the reagent. So therefore I would say a therapeutic approach that has future in the treatment of bladder cancer is a target therapeutic approach. So something that somehow it manages to bind to the tumor cells. So that's where we come in with our expertise in ligand binding, and we have designed many different kind of agents, including nanoparticles, which are able to target tumor cells. And in order to do this research, not only we work in vitro, but also we work in animal models, including dogs and mice. And here you're just seeing a couple of flashy pictures showing some of our nanoparticles targeting bladder cancer cells. Also, we're very interested in how these endocytic proteins recognize the receptors, and in particular, we have been working on understanding how these elements are able to recognize ubiquitinated proteins of the plasma membrane, specifically some of them, but not others. And I hope you're going to be able to read about this very soon in the literature. But we also been able to develop bioinformatic approaches to try to understand how these proteins, what do they recognize, what kind of signals they recognize in cargos present in the plasma membrane that need to be internalized. But what I'm going to tell you today mostly about is this connection with the regulation of Rho GTPases through the interaction with Rho gaps and with Epsin. And this has to do, as I said, with cell division, cancer, and developmental diseases. So these regulators are important for cell polarity, as we said, and cell polarity in particularly with respect to cell migration and cell division. And this was a project that we initiated in Saccharomyces cerevisiae. And here you can see a movie in which it shows Saccharomyces cerevisiae budding yeast, actually budding. So what you see is our normal cells grow in a bud until the bud is similar in size to the mother, and then they separate. So this is the normal behavior of the cells, and it occurs upon Epsin normal function. But if we introduce a point mutation in Epsin, we can generate an exacerbated function of Epsin, an uncontrolled function, and this is the result. What you're seeing here is an hyperpolarization of the bud, so once again connected to cell polarity, and on top of that, what you're seeing is the lack of cell division. You can see in many cases that the mother cell is growing a bud, and without separation from the mother, this new bud grows another bud on top of it. So that's a very abnormal process. So all this just because we introduce a point mutation in an endocytic protein. We then also obtain evidence that when we interfere with the Epsin function, what we get is this kind of phenotype you're seeing here towards the right, which is You can see cells observed by DIC microscopy, and you're seeing gigantic cells in the sample, which are cells in which cell polarity have been completely abolished. Therefore, the delivery of vesicles for growth, it cannot be done towards the bud, but it's done against the whole surface of the cell. Therefore, it leads to the creation of gigantic cells. And this we could determine that indeed was due to raw GTPase signaling imbalance. What you're seeing here in this Western blot out of pulling down activated CDC42 is that in the presence of the point mutation in epsin that I referred to before, the activation of CDC42 that is bound to GTP has been decreased, and it's about 38% what you find in normal cells. So definitely this connection is important for controlling cell polarity. Definitely that's shown in yeast, but now we wanted to know how that affects the cell polarity process in mammalian cells. And what we did is to overexpress epsin first, and you can see that in this picture showing epithelial cells. You know, epithelial cells are all tight junction linked. They don't move, they don't walk away. But when you overexpress epsin, and here you can see it by the expression of epsin GFP, you see this green color associated with the cell that you see towards the right. The morphology changes of the cell and becomes mesenchyme in shape. And actually, it gains the ability to migrate. And in other words, it mimics an invasive metastasis-like behavior. And then this is actually very interesting because, first of all, if you see this kind of behavior in cancer cells, in which epithelial cells become malignant, becomes invasive by producing this change, the epithelial to mesenchyme transition. And the interesting thing is that if we inhibit the function of epsin by using, for example, siRNAs, what you have are cells that can no longer invade. And that's what you're seeing here in the movie towards the right, in which the cells that have been knocked down for epsin function, they land on the substrate, but they can no longer move. And you see them bleeding in the lower part of the movie, but actually that is not apoptosis, it's actually hyperactivation of RAW as a product of the lack of activation of CDC42 and RAC. And that's what we see as a signaling imbalance as well when we do a similar experiment to the one I described in yeast. Upon a siRNA knockdown of epsin 1 and 2, you see the RAC, activated RAC1 being lower, and we also can detect that lack of activation by using FRET biosensors that sense in the living cells when the activation occurs. And then what you see that the epsin double knockdown is definitely not showing any sign of the activation of the FRET biosensor. So we have a connection with the disease of this raw GTPase signaling connection of this endocytic machinery, and as I mentioned, is in cancer. Epsin have been seen upregulated in cancer, and we think it doesn't make the cells malignant, it makes them invasive, it makes them migratory. But also, we've been able to find a disease in which not only is the function is upregulated, but actually a disease in which the function of this signaling circuit is impaired. And the RAW gap that actually involves interaction with epsin is called OCRL1. So OCRL1 is, besides being a gap, although it's an inactive gap, it can bind to raw GTPases, but it doesn't induce the GTPase hydrolysis. But it also is a phosphatase molecule with a specificity, a lipid phosphatase with a specificity towards PIP2. As you know, it's a lipid that is highly abundant in endocytic sites and also on sites of polarized growth. And actually, when this OCRL gene is mutated, it gives origin to a terrible disease that is called OCRL, or oculocerebrorenal syndrome of Lowe, or just Lowe syndrome. It was first described in the early 50s, and the gene was identified in the early 90s. And as I said, it's a lipid phosphatase. It produces a triad of symptoms, which is mental retardation, cataracts in both eyes at birth, The babies are very easily recognized for the cataracts at birth, and also renal dysfunction that is detectable very early on, within a month of birth. You can see the composition of the urine is affected, and it gets only worse with age. And unfortunately, it leads to the premature death of these children after adolescence. The mechanism, although we know the gene and we know what the gene is good for, the mechanism of how to trigger this disease is uncertain. So based on our previous discovery, we postulated that this should be important for cell polarity as well. And considering what we observed with Epstein, it should be involving cell migration at the same time. And here you can see some light imaging done on fibroblasts obtained from the skin of normal individuals and also from two independent non-related patients having Lowe syndrome, LS1 and LS2. And what you're seeing is a movie showing the migrations towards the right of these cells. And also you see the traces of the migration as the computers follows them and calculate the velocity and persistence of migration. And as you can see, the Lowe syndrome cells are slower and they have a low persistence of migration. And you can see that many of the cells, the trajectories are not going forward, but they are going sideways, and sometimes they're going backwards. We did different kind of assays to verify this phenotype in patients, like scratch assays, and we could verify them. Also, we did some migration in transwells, and we could verify the phenotype as well. Actually, we even identify another phenotype, which was cell spreading. If you imagine, this is a very simple test. You lift some cells in suspension, sit them on a surface, for example, fibronectin coated. The cell lands, adheres, and then spreads in every direction. And if you look at them from the top, and that's what the cartoon represents, you can see the nucleus in the center and the cell is spreading in all directions, and it looks like a sort of a fried egg. And here you can see the spreading process using normal cells. You can see again the fried egg with the nucleus in the center. And if you do the same with Lowe syndrome patient cells, this is what you see. And again, as I always said, you don't need to be an expert in cell spreading to tell that the patient cells are really cannot really do what the normal cells are doing. So they cannot sustain the spreading. It seems to be collapsing all the time. And here is just light imaging of the actin cytoskeleton as this process occurs. So essentially what I'm telling you here is that Lowe syndrome patients' cells display defects in cell migration and in cell spreading. And why is this important? Well, this is important because, as you know very well, these processes are very important during embryo formation, during embryogenesis, organogenesis. So we think that this is the cause for some of the developmental defects that we observe. We actually created, in collaboration with Martin Lowe, a colleague in the UK, and Phil Lowe in the UK as well, a system in which we use zebrafish and we use morpholinos initially to inhibit the function of OCRL. And we observe also cell migration defects in the neural crest cells. And as you see, there's a problem with the differentiation of the cells, the migration of the progenitors to produce the cartilage of the face of the fish. So we think we have observed a mechanism for these developmental defects. So that's what I was telling you about how this relates to cell spreading and migration. And we actually look at the raw GTPase, following again the same ideas that we saw with Epstein. And actually we saw similar results. We saw the RAC is poorly activated in Lowe syndrome patients, consistent with this signaling circuit being less functional. And other colleagues have been able to demonstrate that as a response to a lack in activation of RAC1, we have a hyperactivation of RAW-A. So we think definitely this is a disease that results from a raw GTPase signaling imbalance. Nevertheless, in recent times, we've been able to demonstrate that there is also another component to the Lowe syndrome, which is a primary cilia defect. So, and this is a picture that shows actually 4 patient cells. The 2 in the corners have not been transfected with GFP-OCRL wild type, but the ones in the center have been transfected with GFP-OCRL wild type. And as you can see, the primary cilia, here noted because have been stained with acetylated tubulin in red, are only present in the cells that do contain wild type OCRL. The 2 in the corners do not have it. They cannot assemble primary cilia. And we can see that in vivo as well. Our morphant Lowe syndrome animal model shows a problem in the cilia in pronephros. Is very deorganized. So we definitely propose a model in which OCRL participates of the trafficking of proteins towards the nascent primary cilia. Defects in OCRL function lead to defects in the assembly of primary cilia. And we could demonstrate that there is a compartment down just below the base of the primary cilia in which OCRL1 colocalizes with cargo. Here is this chimera called TAC-VXP. VXP is a ciliary localization signal, and it localizes with one interaction protein for OCRL1, which is called IPIP27A. So this is a compartment that exists at the base of the cilia. So since then, now Lowe syndrome have been called a ciliopathy or a ciliopathy-associated disease, and these findings have been corroborated by multiple other investigators in many different parts of the world. Why is this important? Well, it's important because ciliopathies is a very populated area of research. There are new therapeutics opportunities surfacing or being tried pretty much every day. And we hope that we can utilize some of these advances to actually adapt them or adopt them for Lowe syndrome treatment. And so what I've been telling you so far is that Lowe syndrome patients present problems in cell spreading and migration, but also in primary cilia defects. So these were kind of the first cellular readouts for Lowe syndrome, and we decided that they could be used to screen for compounds that might revert these phenotypes, and we did that. And so we actually look for compounds that could, for example, rescue the cell-spreading phenotypes. Here you can see again the cells, the Lowe syndrome cells with difficulty to spread. To the right, you can see some histograms actually showing that the size, the area of the Lowe syndrome cells is prone to be smaller than the area of the normal cells because of this spreading defect. And we identify a drug that we call candidate 1 for now that have been able to rescue the spreading phenotype of these patient cells. And not only rescues the spreading, but it can rescue the activation, the fluid phase uptake is another phenotype that I didn't refer to. It also depends on Rho activation, RAC and Rho activation, and is affected in these patients. It can be rescued with this candidate drug, and it also can rescue the activation of RAC. So obviously we immediately asked whether this candidate number 1 was able to rescue the primary cilia defect, and unfortunately the answer is no, it cannot. So then what we decided is to look for drugs that could suppress the primary cilia phenotype. And we did obtain a candidate called candidate number 3, which was able to reestablish the length of the primary cilia in cells, and was also able to rescue the cartilage problems in the face of this fish. So it is a successful candidate to rescue the primary cilia defects. And then we asked whether it was able to rescue the spreading phenotype, but unfortunately the answer was also no. And this actually led us to believe that OCRL has a dual function, acting in 2 different signaling pathways. Some of them are rescued by a candidate drug, and the other pathway by a different candidate drug. So we think in cases in which both phenotypes are present in patients, combined therapy is what needs to be done. And with that, for that, we are collaborating with colleagues in Northwestern and Indiana University to create the first clinical trials in this respect. What we are now currently exploring is the possibility also of doing cell replacement therapy, and for that, we were the first ones to take fibroblasts from the skin of these patients and create induced pluripotent stem cells. Here you can see the markers of stem cells, TRA160 in particular, the creation of these colonies out of fibroblasts. And we've been able to reprogram them or differentiate them as renal cells, and some of the markers are shown here. And we've been able to show that these cells do show the same kind of primary cilia defects that we've seen in fibroblasts, either in the reprogrammed cells or also in knockout HK2 cells. But something very interesting that we've seen when we induce the differentiation of these cells in the iPSC cells into renal cells is that the very important transcription factor called 6-2, that as any transcription factor, relocalizes to the nucleus. And you can see that in the upper row. I ask you to focus on the upper row of images that you see on the third column. You can see 6-2 labeled in red, and it's showing its localization in the nucleus. If you go to the row below that, what you're going to see is Lowe syndrome cells. And the red arrowheads point out to nuclei that is actually lacking 6-2. Interestingly, the 6-2 seems to be localized or mislocalized to the Golgi apparatus. And actually, that's a place where OCRL localizes, so we think it's connected. And in fact, we suspected that this mislocalization of 6-2 outside of the nucleus, it could be related to problems with the phosphatase activity of Lowe syndrome, and therefore we use lithium chloride, which is the lower of the rows, and you can see in the 6-2 column that 6-2 is again localized at the nuclei upon addition of lithium chloride. Lithium chloride, what it does, it inhibits the synthesis of PIP2, the substrate of OCRL1. So what we are seeing here is a critical transcription factor that it cannot reach its target in patient cells, not at least efficiently, and therefore we speculate that this will produce problems for the generation of all the cellular lineages that depends on 6-2 during the development of the kidneys. And along the lineages affected will be the proximal tubule cells, which are actually known to be affected in these patients. And what you see towards the right are just quantification of the effects I just described for you. So, and actually we do see an imbalance in the generation of different lineages in these kidney cells in vitro. We see, for example, CKA positive cells, which are representatives of the duct, which are overrepresented in Lowe syndrome as opposed to normal cells. And that's the reason is because 6-2, it cannot fulfill its function. Therefore, these cells are then induced to differentiate into a different lineage, in duct lineages. And we could reproduce this phenotype of 6-2 mislocalization by using, again, OCRL knockout cells, HK2 kidney cells. So next step in our work is to prepare kidney organoids, and we actually, I'm showing you here a picture of one of our first kidney organoids and stained by the different different cellular types that make the nephrons. So we are very excited about this and trying to be able to recapitulate the production of a kidney in vitro and study the defects that the Lowe syndrome imposes into the disorganogenesis process. And with that, I'm going to finish, not before acknowledging First of all, the people in the lab that participated in these processes, and all the process of developing these different lines of research. We probably summarize about 10 years of research in this presentation. Brian Kuhn was one of the persons that initiated this research, was followed by Kyle Mathivanan. Both of them are successful postdocs in Yale and in Scripps, respectively. And then the people in the lab that continue their research, Claudia Hanna, Wenjie Xie, Shweta Ramadesikan, Makit Pearson, and we have our undergrad helpers, Chinma and Brittany. And Shweta and Wenjie have been very instrumental for the development of what I just described today, particularly Wenjie for the iPSC and the reprogramming that I described, and Shweta for the identification and characterization of the candidate drugs for counteract the effects of Lowe syndrome. And then my collaborators in the UK, Phil Beals and Martin Lowe, Donna Fickety in our Institute for Integrative Neuroscience, and Phil Lau in the Drug Discovery Center. These two colleagues at Purdue have been instrumental, Donna for the iPSC story, and Phil for continuing the developing of therapeutic approaches against Lowe syndrome. And what you can see down there is our funding agencies, NIH and NSF, but also I want to highlight the Lowe Syndrome Trust, which gave us funds to initiate all this research, and the Lowe Syndrome Association as well, and now is continuing to fund us, as well as the Indiana Clinical and Translational Science Institute. Recently, they have given us money to continue with this research. And without that, I will just finish and take any questions if you have it.
I would like to thank Dr. Aguilar for his presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through December of 2018. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's event. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.